this means electron transport system. So what is actually electron transport system? What is the aim of this electron transport system? The aim of electron transport system is that to release energy. That energy is stored in the NADH2 and FADH2. NADH2, nicotinamide adenine dinucleotide, and FADH2. The energy stored in the NADH2 and FADH2 is released during this electron transport system. So that is the main aim of this electron transport system. NADH2 and FADH2 is forming during the respiration, both aerobic and anaerobic. Glycolysis and Krebs cycle, this NADH2 and FADH2 is forming. The energy have to release from the NADH2 and FADH2. That is the aim of this electron transport system. So during the electron transport system, the energy present in the NADH2 and FADH2 is released by the oxidation of these two molecules. And finally, the electron is transferred to the molecular oxygen, and molecular oxygen undergoing reduction or the ionization, and this oxygen combines with the hydrogen and forming the water molecule. That water molecule is known as the metabolic water. So this electron transport system is actually taken place in the mitochondria. In the mitochondria, the inner wall, the outer side of the inner wall, that is the location of this electron transport system. So what are the electron carriers during this electron transport system? Electron is transporting from one carrier to another carrier, and finally this electron is giving to the oxygen, and oxygen become ionized, and then water molecule is forming. So what are the electron carriers present in the electron transport system? I will take a few electron carriers. So already these electron carriers are shown in this diagram. So one of the electron carrier is the FMN. FMN means flavin mononucleotide. This is one of the electron carrier. Another electron carrier is the coenzyme Q, or it is called a ubiquinone, or it is called the coenzyme Q. And third electron carrier is the sulfur-containing ion compound, and this sulfur-containing ion compound helping to transfer the electrons from one carrier to the another carrier. Then another electron carrier is the cytochrome, iron-containing cytochrome, different type of cytochrome, that is cytochrome B, C, C1, C, and A3. These are the different types of cytochromes. All these are the iron-containing electron carriers. So these are the different types of electron carriers. Then another important factor you should understand, that is this electron transport system having total 5 complexes. So what are the different complexes? What are the aim of these different complexes? Complex number 1. Complex number 1 is known as the NADH dehydrogenase. NADH dehydrogenase. This is the first complex, complex number 1. Complex number 1, NADH dehydrogenase. It is already shown here. This is the NADH dehydrogenase. That is the complex number 1. And the complex number 2 is related with succinate. It is called a FADH. Complex number 2. It is called a FADH dehydrogenase. Or it is also known as the succinate dehydrogenase. Succinate dehydrogenase. And the complex number 3 is related with this one. It is known as cytochrome B C1. So cytochrome complex 3. It is called a cytochrome B C1 complex. That is the third complex. And fourth complex is coming here. This is the fourth complex, and this fourth complex involves cytochrome A3. So that forming it is called cytochrome oxidase complex. Cytochrome oxidase, and this cytochrome oxidase, or the fourth complex, third complex, and second complex, first complex is coupled with the fifth complex. That fifth complex is present here. That is known as the ATP synthase complex. ATP synthase complex, or it is called a ATPase complex. So like that there are 5 different complexes, and these 5 different complexes having inhibitors. So by using this inhibitor we can identify these different complexes. For example, the complex number 1, NADH dehydrogenase, it is actually inhibited by the retinol and amytal. Retinol and amytal. There are 2 chemical compounds, that is retinol and amytal. It will inhibit this first complex. And second, the complex number second, it is not actually inhibiting by any antibiotics or any other chemicals. And the complex third is known as cytochrome BC1. It is inhibited by antimycin and antibiotics, antimycin. And the complex number fourth, cytochrome AA3, it is inhibited by cyanide and carbon monoxide. Both cyanide and carbon monoxide, you know that it is highly poisonous, and this poison inhibits the cytochrome AA3. And the fifth complex, that is ATP synthase complex, and this complex is inhibited by the oligomycin antibiotics. So by using these antibiotics, we can identify the different complexes present in the electron transport system. So these are the 5 different complexes in the electron transport system. Then say, what is the role of the oxygen in the electron transport system? We know that oxygen helping for the complete oxidation of the glucose, and finally we will get 38 ATP molecule. So where is the oxygen? After this, in the glycolysis or in the Krebs cycle, there is no oxygen directly participated. And here also, where is the oxygen? The oxygen present at the terminal reaction. This reaction, oxygen present here. So this oxygen, actually the electrons transfer from carrier to carrier to carrier, and finally the oxygen undergoing ionization or the reduction and combines with the hydrogen and forming this water molecule. This point, that is known as the terminal oxidation. If a terminal oxidation not taken place, the entire system, ETS will stop. If ETS stop, the Krebs cycle will stop. If a Krebs cycle stop, the glycolysis will stop. The total respiratory system will, respiratory process will stop. So oxygen is very essential. This oxygen removing the hydrogen. If hydrogen is not removed, the entire system respiratory process it will stop. So to drive the respiratory process, removing the hydrogen is very essential, and that process is performed by the oxygen. That is the role of the oxygen. So, and cyanide, I told cyanide will inhibit the complex of fourth. So actually cyanide inhibiting at which stage? It actually inhibiting cytochrome oxidase. Cytochrome oxidase. Cytochrome oxidase. It is an enzyme. This cytochrome oxidase is helping for the oxidation of the A3. It is helping for the oxidation of the A3. At the same time, it is helping for the reduction of the oxygen. Oxygen become O minus. These two processes will stop if cyanide is there. So if cyanide is applied, the entire respiratory process will stop because it is affecting on the cytochrome oxidase. Means the terminal oxidation process will stop. That means the entire respiration, Krebs cycle, glycolysis, and all these will stop. So that is very dangerous, and within a fraction of a second, this cyanide will kill the animal. It is because of this process. But there are some plants. These plants can perform the alternative route if a cyanide is present there. For example, spinach, a plant, spinach and pea plant or Pisum sativum, pea plant. These two plants are a simple example. There are many plants. Two examples, spinach and pea plants. If a cyanide is present, it take an alternative route. So these plants are known as the cyanide resistant respiration. The process respiration taken place in this plant is known as the cyanide resistant respiration. Cyanide resistant respiration. Cyanide resistant respiration. Respiration taken place in the pea plant and the spinach plant. And also it is known as the alternative electron transport system because it take an alternative route to complete this, and the plant is not affecting the cyanide. So in the presence of the cyanide also, this plant will live successfully. Most of the plants cannot live because the A3, the terminal oxidation process will stop. So that is the background of this electron transport system. So now see how the electron transport system taken place. So in the electron transport system, NADH, the energy present in the NADH and FADH2 is releasing. So where the NADH2 and FADH2 is forming in the living cells, NADH2 and FADH2 is forming during the glycolysis and also during the Krebs cycle. Tricarboxyl TCA cycle, tricarboxylic acid cycle or Krebs cycle, this NADH2 and FADH2 is forming. The NADH2 FADH2 is known as assimilatory power because it is containing large quantity of the energy. So the aim of this ETS is to release that energy. So how the energy is releasing? So this is the mitochondrial part. The diagram is representing the mitochondrial part. The central part, this one is called the inner membrane. Mitochondria have two membranes, outer membrane and inner membrane. This is the inner membrane. And outer membrane present here. Between the outer membrane and inner membrane, there is a space that is known as the intermembrane space. Then, after the inner membranes, the matrix will start. So this is the matrix part. That is actually the central part of the mitochondria. So this is the matrix part, and this is the inner membrane, and this is the space between the inner membrane and outer membrane. So that is called interspace. So this NADH is actually FADH and NADH is actually present in the matrix. So this NADH, with the help of the NADH dehydrogenase, it undergoing the reduction and the hydrogen is releasing. So NADH converting into oxidized form NAD, and this hydrogen is released, and this hydrogen is actually split into hydrogen ions and electrons. This hydrogen ions is transporting into the inner membrane. When the hydrogen ion transport, not 2 hydrogen coming, actually 4 hydrogen is transporting into the intermembrane space. And the electron, what happens to this electron? Actually, directly it cannot transport it to the intermembrane space. First, the hydrogen is accepted by the FMN. From the FMN, and a sulfur containing iron, and here it is split, and the electron is transferring, and hydrogen ions are transferred to the intermembrane space. What happens for this electron? This electron is accepted by a ubiquinone complex, that is the coenzyme Q, and this ubiquinone become reduced, it become ubiquinone H. So this hydrogen is taken again from the matrix side. So matrix side hydrogen is continuously decreasing. So it become ubiquinone reduced, and this ubiquinone reduced again split the hydrogen, and electrons are released, and the hydrogen is transferred to the intermembrane space. Again take 4 hydrogen from the matrix, and the 4 hydrogen is transferring to the intermembrane space, and this electron is accepted by the third complex, not a second complex, it is entirely different. Third complex, cytochrome B, and the cytochrome B transfer the electrons to the ubiquinone, and sulfur containing iron complex, and this transfer the electrons to the cytochrome C1. So these two forming actual cytochrome B C1, that forming the complex number 3. So from the cytochrome C1, the electron is giving to the cytochrome C. So see here, the cytochrome C and ubiquinone both are very peculiar because these are the mobile electron carriers. It can move from place to place. Ubiquinone moving from place to place, and the cytochrome C also moving from one place to another place. So cytochrome C and ubiquinone is commonly known as the mobile electron carrier. So now the electron is present from the cytochrome B. Electron is present in the cytochrome C. So this cytochrome C is moving downward, and it is reaching to the copper containing, that is the 4th complex. This is the 4th complex. 4th complex containing copper, central copper. There are 2 copper, 1 copper here. So this electron is giving to the copper, and then it is moving to the original position. So when this happens, actually there are 4 electrons, 2 electrons. Both electrons at a time cannot transfer. First 1 electron come, and it is giving to the copper, and from the copper it is moving to the cytochrome A, from the cytochrome A to the cytochrome A3, and then to the another copper. From this copper, this electron is giving to the molecular oxygen, and the molecular oxygen become ionized, and then combines with the hydrogen and forming the water molecule. Okay, then 1 more electron is there, and this electron again come downward, and it is transferred to the cytochrome A3, and with the help of this energy present in the electron, 2 hydrogen ions are transferring to the interspace. Actually here 4 hydrogen are transferred because 2 electrons having that much energy, and 4 hydrogen is transferred. Here also 2 electrons have that much energy, and 4 hydrogen is transferred. Here only 1 electron used for the ionization of the oxygen, and with the help of the 1 electron, the energy is very less, and it transport only 2 hydrogen to this side. So like that, on the matrix side, the hydrogen amount is reducing step by step, and similarly on this side, the hydrogen ions amount is increasing little by little, and as a result a proton gradient is forming. So this is here more protons, here less protons, so proton gradient is forming. Along with this, the energy electrochemical gradient also forming here. This side more hydrogen, more positive, and this side less negative. So due to this electrochemical gradient and this proton gradient, a force is applying there, that is known as the proton motive force. Proton motive force. Proton motive PMF, proton not electron, proton motive force is set up, and this proton motive force cannot allow the hydrogen to remaining on this side, and due to this force the hydrogen must be transferred to the next side. When the hydrogen is transferring to the next side, the fifth complex is coming there. The fifth complex, I told you, it is a ATP synthase or ATP synthesizing enzyme. That enzyme is known as ATP synthase. This we already studied in the chemiosmotic hypothesis. It has a two part, that is a F0 part and F1 part. This hydrogen is moving through this. So there are total 10 hydrogen is formed here. So 4 plus 2, 10 hydrogen is formed. This is the latest, in the latest edition of the NCERT book, this diagram is given. In the old edition, the diagram showing different types arrangement, and here only 2 hydrogen, and 2 hydrogen. Total 6 hydrogen formation is shown in the old textbook. And the new textbook, the colorful diagram with the 4 hydrogen, and 2 hydrogen, that is a total 10 hydrogen is shown. So out of the 10 hydrogen, first 4 hydrogen is moving through this F0, and then conformational change on this F1 part, and here energy is created, and this energy utilized for the synthesis of the ATP molecule. So core hydrogen is hydrogen ions requiring for synthesis of 1 ATP molecule. So total 10 hydrogen, out of the 10 hydrogen, total 10 hydrogen is formed. So 10 divided by 4, that is the 2.5 ATP molecules. So when 2 hydrogen moving through this ATP synthase, 1 ATP is forming. So 6 upon 2, that is equal to 3. Like that, the old textbook is strong, but according to the latest, 10 hydrogen is forming. Okay, so like that, FADH. So now we discussed only about the NADH. What happens for the FADH2? FADH2 forming or the FADH dehydrogenase, succinate dehydrogenase complex. That complex is the second complex. The second complex is not involved in the NADH, the energy releasing from the NADH. So from the FADH we have to release energy, then the second complex is coming. Second complex succinate converting into the fumarate, then ATP is forming. Okay, so here hydrogen is FADH forming, not ATP. FADH forming. This FADH become reduced, and FAD transfer the electrons to the ion containing sulfur containing ion complex, then to the ubiquinone. Ubiquinone become reduced, and for the reduction hydrogen is taken from the matrix. Again hydrogen is become less in the matrix, and it is split. Electron is giving to the cytochrome B, and hydrogen is transferred to the intermembrane space, and from the cytochrome B, sulfur containing ion, cytochrome C1, cytochrome C, copper containing central part, cytochrome A3, and again copper, and it is moving there. So this is the another cycle. This is only happening in the FADH. So in the FADH, this part is totally absent. So automatically one ATP is less. FADH giving one FADH giving only 2 ATP molecule, one NADH giving 3 ATP molecule, because the NADH is starting from this point. So it is giving 2.5 ATP, means 3 ATP molecules, and this FADH is starting from this point. So this is totally absent. So automatically one ATP is less. So FADH2 providing 2 ATP molecule, and NADH2 providing 3 ATP molecules. So that is the electron transport system. Then one important point to be remembered here, the FADH, NADH2 is forming in the glycolysis also. Glycolysis is taken place in the cytoplasm. So from the cytoplasm, how the NADH2 transfer to the mitochondria? For the transferring the NADH2 from the cytoplasm to the mitochondria, it is requiring energy. So 2 molecule ATP is utilizing there. So total 38 ATP molecules are not forming actually in the practically, because the 2 ATP molecule is already used for the transfer of the NADH2 to the mitochondria. So instead of the 38, 36 ATP molecules will form. And for the transfer of this NADH2 to the mitochondria, 2 shuttle is using. 2 shuttle processor. What are the 2 shuttle processor? One of the shuttle processor is the glycerol 3-phosphate shuttle. Glycerol 3-phosphate shuttle, GPS, glycerol 3-phosphate shuttle. This shuttle helping to transfer the NADH from the cytoplasm to the mitochondria. And this glycerol 3-phosphate shuttle is operating in the nerve cells, muscle cells, and all the plants. all the plants. So the shuttle used in all the plants, nerve cells and muscle cells is the glycerol 3-phosphate shuttle, GPS shuttle. And another shuttle is the malate shuttle. Malate aspartate mass, malate aspartate shuttle. Malate aspartate shuttle. This shuttle is using, malate aspartate shuttle is using to transfer the NADH from the cytoplasm to the mitochondria in the case of the heart muscle, in the case of the liver, and in the case of the kidney. So in this kidney, liver and heart, malate aspartate shuttle is operating to transfer the NADH to this. So that is the electron transport system in brief.